In this set of lessons, we'll look at the biological basis of learning and memory. The consensus view in the neurosciences is that synaptic plasticity is the key mechanism for learning and memory. The idea that there are biological changes that underlie learning and memory goes back a long time, all the way back to the pioneering work of Ramon y Cajal, who hypothesized that the bumps on the dendrites of a neuron were connection points with other neurons, where changes associated with learning might occur. And of course, today there is much evidence that Cajal was correct. Learning and memory are rooted in changes at the synapse. Now, if we take a look over here at some of his fantastic drawings here, we see two neurons here, the cell body, axon. We got an extensive dendritic tree, and you can kind of see the little bumps on the dendrites here. Those would be spines. What we don't see are the axons and terminals making synapses on those spines. But Cajal was an expert microscopist and neuroanatomist, and he speculated that learning involves biological changes at certain sites on the dendrites of neurons. Decades later, in the mid-1900s, scientists were gathering more evidence that learning involves physical changes in the brain. Take one group of rats and raise them in an impoverished environment, another group of rats, raise them in an enriched environment, and then look at the brains of those rats later on, you find significant differences. In rats in the enriched environment, you have a thicker cortex, larger cells, more extensive dendrites, more dendritic spines. It's as if the extra learning that was happening in the enriched environment was causing physical changes to neurons and their interconnections. What extra learning is happening here? Well, you have a lot more social interaction here. There are things to learn about other rats, lots of toys to explore. In the impoverished environment, no social contact and no other toys to learn about. So the suggestion was that learning does indeed involve physical changes in brains. Also in the mid-1900s, a Canadian psychologist by the name of Donald Hebb articulated what is today still sort of the basic understanding of what's happening during a learning process. He said that when axon of cell A is near enough to excite cell B, or repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficiency as one of the cells firing B is increased. Today, it's often summarized as cells that fire together, wire together. So at this point then, we can start to put some pieces together. We know from the case studies of HM and others, the hippocampus seemed to be a critical structure in forming memories. And at the same time, it was understood that perhaps physical changes were happening between neurons in the brain, and this accounted for memory. We'll call those physical changes at the synapse synaptic plasticity, referring to the capacity of synapses to change their strength. So perhaps what was going on is that when we undergo some kind of learning episode, physical changes, synaptic plasticity is happening between neurons in the hippocampus and other regions, and long-lasting changes then would be the basis of a memory. We can apply the word consolidation here as well. Synaptic consolidation then would be the fundamental process that converts a learning-induced change in synapse strength into a long-term memory. In other words, if you increase the synapse strength and that increase in strength persists for a long time, that then would be the biological basis of a long-term memory. Notice consolidation we learned before in the system context, where system consolidation referred to the reorganization of a memory across different brain systems, but still in both of these systems, the changes that would be happening at the level of neurons would be synaptic plasticity. So we would have synaptic plasticity among hippocampal neurons and synaptic plasticity happening among cortical neurons. So in the remainder of the lesson then, we'll take a look at the hippocampus in more detail. Recall the hippocampus resides underneath the temporal lobe bilaterally. Here's a cross-section of the whole brain showing the location of the hippocampus, and here a section of brain has been stained to see cell bodies. Take a look at higher magnification. We can see some structure of the hippocampus. We have fields of cells, the dentate gyrus and CA3 and CA1 cells. In higher power magnification, we can start to see individual cell bodies. Now, a breakthrough occurred in the latter part of the 20th century, in the late 1900s, where scientists were able to make thin slices of the hippocampus and keep the tissue alive in solution. So the slice would be bathed in a nutrient solution, and you could keep this slice alive for up to 6 hours or so, whereby you could then play around with stimulating and recording electrodes, so you could gain control over the nature of neuron communication within the hippocampus. You could have stimulating electrodes that would stimulate axons to generate action potentials, and you could have recording electrodes either in the population of target cells or recording the electrical activity of one target cell only. So this experimental arrangement led to some fundamental breakthroughs in our understanding of synaptic plasticity. A very important discovery was made in the 70s with hippocampal cells. Let's imagine here are two hippocampal cells, the presynaptic cell and the postsynaptic cell. Now, imagine that in this pathway for the presynaptic cell, we just give a small test pulse. So we activate the cell so that it makes one action potential. Well, it arrives at the terminal, some neurotransmitters are released, and they bind to receptors, and ions will enter, and you'll get some kind of small electrical response, a depolarization in the target cell. Now what they did was, to that same neural pathway there, they stimulated the presynaptic cell at high frequency. So lots of action potentials racing down. We're going to get lots of neurotransmitters released, a lot of channels opened up, and a lot of ions, sodium ions, rushing in, and we will pass threshold, and the target cell will now generate its own action potentials in response. But here was the fundamental discovery. If then, a moment later, you go back and just deliver one test pulse, one action potential down that same neural pathway, what they found was an increased response in the target cell compared to the original cell. Right? So it's as if this high frequency stimulation of this pathway that generated activity in the target cell, it's as if that activation somehow increased the strength of the synapse. And this strengthening of the synapse became known as long-term potentiation, because the enhanced strength of the synapse lasted for many hours. And it was potentiated. The synapse was potentiated. It was increased. Now, what are some of the possible mechanisms of long-term potentiation? Well, you'll recall a synapse has an axon terminal, and when the electrical signal reaches the axon terminal, these little vesicles filled with neurotransmitters fuse to the membrane and dump the neurotransmitters into the gap. They diffuse across the gap. They bind to protein receptors on the spine of the target cell. These are channel proteins that allow ions to rush in, and that's an electrical signal. So that's how a synapse works. Now, this immediately presented a couple possibilities. Maybe what happens when LTP is induced is that maybe the target cell inserts more channels, more receptors into the membrane here. So for a given amount of neurotransmitter, you'll have more open channels, more ions can go in, you'll have a bigger electrical signal in the target cell. Or on the other hand, for each electrical signal reaching the terminal, maybe more chemicals are released, more transmitters are released, and with more neurotransmitters, you can keep these channels open for longer, and you'll get a bigger electrical signal in the target cell. So these were two possibilities. And in fact, there is solid evidence that the target cell does indeed insert more protein channels in the membrane here. So for a given amount of neurotransmitter released, you're going to have more current entering, more electrical current entering the target cell. And there is solid evidence that long-lasting changes at the synapse involve structural changes to the synapse. So if the spine used to have this shape, that's the old spine, over the course of hours and days, what you find is a larger spine. And with a larger spine, the cell can insert more receptors. Again, the effect of that will be to generate a larger electrical signal in the target cell for a given amount of transmitter released. So we can classify these changes based on the time scale. We can call the early changes, the ones that happen rather quickly, functional changes. The synapse, the strength of the synapse is increased quickly by quickly inserting extra receptors into the target cell membrane. And then there are long-lasting changes. We'll call them structural. These are the kinds of changes that involve changing the structure, actually building a larger spine, for example, and a larger synaptic junction. And these kinds of structural changes may underlie the long-term changes at a synapse that would be the basis for long-term memory. So the strength of neuron communication can change, and this is called synaptic plasticity. Now, we've just seen the example of the synapse getting stronger, but there's evidence that synapses can also get weaker. So synapse strength can change in both directions as a result of experience. Changes at the synapse involve biological processes. Proteins are being produced, for example, in the spine, and perhaps the receptors are inserted into the membrane, and that would strengthen the synapse because you'll have more electrical current getting into the target cell. And these receptors can be removed as well, and that would be a case of the synapse getting weaker. Structural changes in the dendrite shape, in the shape of the spine, seem to persist over long periods of time, and this may be the basis for maintaining these synapse changes. So synaptic plasticity is the biological basis for learning and memory. During a learning episode, we get a synapse strength change, and the memory then would be that synapse change is maintained over time. And this means that the information that was learned can be retrieved for use in our ongoing life. After establishing the basic phenomenon of LTP, long-term potentiation, scientists went on to characterize the conditions under which LTP occurred at a given synapse. There are three conditions here that we'll take a look at, and then we'll draw some conclusions. Under the first scenario, what scientists did was they stimulated with high-frequency action potentials the presynaptic cell, but while they were doing that, they prevented the target cell from generating action potential. So they hyperpolarized it. They injected chloride ions, negative current into the cell, to prevent it from responding. Under these conditions, you do not get the LTP phenomenon. You do not strengthen the synapse. It's as if the target cell must fire in order for a synapse to be strengthened. In scenario 2, we stimulate the presynaptic cell again with high-frequency action potentials, but it's a weak pathway. It would be insufficient to drive the target cell to make its own action potential, so the cell won't reach threshold with this stimulation, but we come along and we inject extra current, so we can reach threshold now. When a weak pathway is stimulated and we stimulate the target cell, yes, indeed, you do get LTP. You can strengthen this weak synapse. Condition 3, if we simply stimulate the target cell by itself without stimulating the presynaptic terminal, you do not get LTP. In other words, this suggests that you need the presynaptic terminal to be actively taking part in firing the target cell for LTP to happen. In other words, if you take 1, 2, and 3 together, this is support for Donald Hebb's suggestion that neurons that fire together wire together.